New research is looking at the growth and development of brain cells to better understand how to help those affected by neurological conditions, which are a leading cause of illness and disability worldwide. Joining us now is Madeline Anders from ASU's Ira A. Fulton Schools of Engineering. Welcome to Arizona Horizon. Good to have you here.
Thank you so much for having me.
All right, your research, this is really interesting. I think I got it right here. Your research is like guiding cells outside of the brain to become brain cells?
Yes, absolutely. So there's different ways that you can study the brain, but obviously we can't go into someone's brain that's alive and look at how their cells grow and change in the context of neurological diseases. And so what my lab does is we take blood or skin cells, often samples that are taken from kind of routine clinical assays, and then we can take them and reprogram them until they're in this sort of developmental state, and then study how they change over time. And so this provides a really new tool and resource to really look at how different neurological disorders arise and maybe some ways that we can treat them.
Are these like stem cells you get from skin and
yes,
that kind of stuff?
Exactly. So basically there is, this actually won the Nobel Prize back in 2012. There are strategies now to take cells that are from mature tissues like skin or blood and then take them and push them sort of back to an early developmental stage. These are called induced pluripotent stem cells. And then when they're in this early pluripotent stem cell stage, you can push them into any cell type of interest in the body. And we're interested in the brain, and so we push them toward this brain-like cell.
So now that you've pushed them in
Yes,
that direction,
yes.
what are you finding?
Absolutely. So we are really interested in understanding how they change over time, both in sort of healthy development and in different disease states. And what we are really excited about and interested in pursuing moving forward is really understanding how nutrients in the brain, how we consume nutrients, and then how those things are processed to produce energy, how that becomes disrupted in different neurological disease states. And so we know from a variety of different both neurodevelopmental and neurodegenerative disorders that there's alterations in these metabolic programs. And so we're really interested in studying how these things change in sort of healthy development and also in alterations in different neurological disorders.
Basically how the brain uses energy.
Exactly.
Okay.
Exactly.
How important, because I read about this stuff here, the best as I can understand it here, it sounds like glucose is a biggie.
Yes, yes. So glucose is the main energy fuel in the brain, and actually when we eat various calories, right? Glucose is basically 20 or 25% of the glucose is consumed into the body is utilized by the nervous system or the brain. And so it's really, really important for how we break these fuel sources down into producing energy. And when these programs become disrupted, it can disrupt how the cells within the brain communicate. And so the cell culture model systems that we utilize, we're trying to study how this happens normally, and also when we disrupt, and because we have access to these cells in a dish, we can manipulate these programs and then see what happens.
But what about things like glucose intolerance? I mean, we have a whole type 2 diabetes endemic out there, it seems like. How does that equation work into all this?
Yeah, so there is some, I think, more whole body kind of understanding of how hyperglycemia can really affect brain function and health in different sort of both development and degenerative disease states. And there is, at least from sort of a public health perspective, there are some correlations there. But really, we're interested in understanding mechanistically how these things can alter function and in the way that the brain cells communicate with one another, and we're trying to understand how this could maybe be a way that we could use it as a sensor to indicate onset of different neurological diseases, or if this could be a target, or if we could prevent some of these changes to prevent onset of different neurological diseases.
So some of these diseases, could it possibly be a factor of having too much glucose, not enough glucose?
Yes,
Either
absolutely.
Or.
Yeah, either or.
So that's a factor. That is whole body then. You really have to kind of put that into the equation.
Yes, absolutely. And so it's really important to understand, I think, the refined level, right? So dysfunction can either be not enough or too much, and so there's sort of a Goldilocks kind of happy medium.
What about other nutrients?
Also very important, we're interested in regulation. So during very early development, glucose metabolism is very important for the way that energy is used and produced. We're also interested in lipids or fats, right? How those are important for brain and body development and function. Cholesterol is also very important. And so there's different sort of strategies that you can use to study different kind of components and how they're broken down and utilized appropriately to both build the brain but also help it function appropriately.
Last question, what's next in this research?
Oh, wow. So we are now using induced pluripotent stem cells that have been derived from patients with different neurological disorders, either autism or Alzheimer's disease, and we're trying to understand how these might be different in these patients.
All right, Madeline Andrews, ASU, this is really fascinating stuff. You're on the cutting edge here of this kind of research. It's good to have you here.
Thank
Thanks
you
for
so
joining
much.
us.
Really appreciate it.
You bet.